Okay, so kidney and osmo regulation. So from the word, we do know that we need to control, again, the water, something to do with water, right? Okay, basically to control how, what is the volume of the urines, okay, the volume of the urines in this process, okay? So no doubt we have to go through the structure of kidney, which is a little bit complicated, okay, the process as well. And the last part, osmo regulations. Basically, we want to maintain a suitable range or the normal range of the water potential. Okay, we do know that if the blood's water potential is low compared to the cells, so I mean if the cell have higher water potential, then water molecule going to leave, okay, the cells into the blood. Therefore, shrinkage, it may interfere with a particular metabolic reaction. The cells will lose the functions. If the blood's water potential is higher, right, compared to the cells, then the water molecule enter into the cells and cause the bursting. So that's why we need to control what we call this a normal range of the water potential. Okay, so now look at this. What we're going to learn in these subtopics. Okay, so number one, we're going to describe the process called deaminations. So D means remove. Aminations here means remove of the amino groups or amine groups, okay, from the amino acids, and outline the formations of the urea in the urea cycle. Another name for urea cycle also called ornithine cycle. But compared to the Krebs cycle and also the Kelvin cycle, ornithine cycle or urea cycle, you just need to know the name, or there is a such thing called urea cycle, full stop. You do not need to know the detail of the cycle. It's unlike the Krebs cycle, you need to know, okay, from this oxaloacetate, it becomes citrates and blah, blah. No need. Here, you just need to know, oh, got such thing called urea cycle. What is the functions? To form urea, full stop. Okay? And then after this, we're going to describe the gross structure of the kidney. Gross structure means the gross anatomy. Means that in terms of what you can see without using the microscope. And we need to know the detailed structure of the nephrons with the associated blood vessel using the photomicrograph and electron micrograph. So it means that microstructure, okay, of the nephrons. Nephron is the functional units of the kidney. Are you clear? It's the functional units of the kidney, so that the kidney will be able to carry out the two processes. What are these two processes? They are ultrafiltrations and selective reabsorption. So we need to describe how the process of ultrafiltration and selective reabsorption involve in the formation of the urines. Okay, and the last one part here, there's something to do with the osmo regulations, which involve few organs here. Hypothalamus, we need to know the role of hypothalamus, as well as the posterior pituitary glands. Okay, hypothalamus, pituitary glands, and the respond. Who respond? The collecting duct is the one that respond to what? ADH. ADH also called vasopressin, or the full names here is called antidiuretic hormone. Antidiuretic means that high volume of urine. Diuretic. Anti means this stop to produce more or high volume of urine, so that our body will conserve more water. And the last part, we're going to look at how we can use a urine analysis to look at how glucose, protein, and ketones appear in the urine, what they indicate. Okay, so basically this will be our topics we're going to learn. It's quite long topics, okay, for kidney and osmo regulations. Okay, so we start with first process called excretion. So when you talk about excretion, so excretion is the process by which metabolic waste is eliminated from an organism. So it must be metabolic waste. It's not the other waste that, like for example, when you eat and then you go to toilets and then you have this the bowel movement, those, or good shit in this case, then it's not metabolic waste. Are you clear? It must come from metabolic. Okay. So this process primarily carried out either in the lung, when you release the CO2, kidney, when you release the nitrogenous waste, like for example, urea, skin, okay, skin, particularly the extra we call this as water, sweat, okay? So through the skin. So in animals, where our main excretory product is the carbon dioxide from the lung and also the nitrogenous waste, for example, ammonia, urea, uric acid, and creatinine. We do know that carbon dioxide is produced continuously by the cells that are respiring aerobically. Okay, have to be careful, guys. CO2. If you look at our cells, CO2 release due to these two processes, link reactions and also the Krebs cycle. And both of them happen in the mitochondria. Are you clear? So means that when the pyruvate decided to enter the mitochondria, so means that it must be aerobic respiration. So CO2 only produced during the aerobic respiration in mammal, okay, and also anaerobic respiration in the yeast cells. Okay, so be careful when you write. Okay, it's not the anaerobic or normal respiration, but it's a respiration, it's aerobic respiration. Okay, so then it reaches the lung and then we can excrete out. We also produce ammonia through the process called deamination process. Okay, we produce ammonia. So how we actually produce ammonia? If you look at this, the general form of the amino acid with the R group, hydrogen atom, COH, this is the amino group. So in our syllabus, we can call it amino group, okay, or amine group. Okay, either amino group or amine group. So when you remove this amine group or amino group, then these amine groups will react with the hydrogens, and then you can see that okay, with the hydrogen atom to form ammonia. Can you see that? NH3, ammonia. The remaining structure here, the remaining structure actually is known as the keto acid. Okay, keto acid. So this keto acid, either we carry our respirations to what we call this oxidize the keto acid, or we convert it to the glucose, then we can store it, or we can convert it into the fat. So basically, amino acid, when you eat or you consume too high the concentration of amino acid, it doesn't make you feel strong, because the extra one will be deaminated. So for example, you go for the gym and you take these protein diets too high in the protein, at the end you can see that deamination take place. It will convert to the glucose and also the fats. So moderate. Okay, don't take too much of protein as well. Okay, or you think that, oh, I will go to gym, I want to make up my muscle, so therefore I take this protein diet. So if too high at the same time, you actually convert the glucose, you're going to store it as a fat as well. Can you see that? Okay, so urea is produced. Okay, you can see that urea, we produce a urea from the ammonium. From ammonia. From ammonia, we get the urea. Later, I will tell you why. Then it produced from the ammonia through the process called urea cycle in the liver. Deamination also take place in the liver. Okay, and the uric acid is made from the breakdown of the purines from the nucleotide. What's purine? We have ATCG, right? Okay, so the G and the A, okay, guanines and also the adenosines. Okay, so they are the purate. Okay, they are the purate. Okay, so C and T, they are pyrimidine. So the food that high in the purine will give us high in uric acid. And if the uric acid too high, the accumulation of uric acid at your joint, at our joint, will form the crystal and you cause the disease called gout. Okay, the gout. So gout is very, very painful. Okay, the gout. Okay. So, and creatinine also is break down the product of the creatine phosphate from the muscle. Now, what is a creatine phosphate? Creatine phosphate is something like ATP. Okay, so creatine phosphate, when we carry out the aerobic activity, require high concentration of oxygen, but your oxygen supply cannot meet the demands for the ATP productions. what will happen here is we break down this creatinine phosphate so that phosphate group can be donated to ADP to form ATP, so that with ATP we can continue to carry out our exercise, and the remaining item here is the creatinine. Are you clear? So creatine phosphate group will be taken by the ADP to form ATP, so that we can continue to have the muscle contraction and running, okay, during these activities. And the remaining one is the creatinine. So creatinines, we can't use it anymore. We're going to excrete out from our body, okay, creatinine. So our focus in terms of the urea now, okay, why we have to look at this urea cycle. So deamination is the removal of the amino group, okay, or amine groups, either called amino group or amine group from a molecule. So the enzyme that catalyzes this reaction is known as a D-Aminates, okay, D-Aminates, and primarily carried out in the liver. So if you take in excess proteins, so deamination is used to break down the amino acids. So the amino group or amine groups will be converted to ammonia. The keto acid, the remaining ones, will enter into the Krebs cycle to continue to be oxidized or to convert into the glucose, okay, or glycogen, or even the fats for the storage. Now, but the problem here is we cannot transport ammonia. We know that we need to excrete out the ammonia. But the problem here is ammonia is very soluble and highly toxic. Okay? Highly toxic. It's about 10,000 more toxic compared to urea. So it means that if you want to transport ammonia in high concentration in our blood from the liver to the kidney, it will cause, okay, the problem and toxicity here. Okay, so therefore we have to convert the ammonia into the urea, and the urea is less soluble and less toxic. So when less toxic, so means that you can carry them in a higher concentration to be excreted out. Okay, so how we can get the urea from the ammonia is through the cycle called Ornithine Cycle or Urea Cycle. Okay, so basically in the liver, ammonia will react with the carbon dioxide. Okay, two ammonia with one carbon dioxide. You can see that you're going to form the urea with the formula CONH2. Are you clear? So it's something like this: a CO, NH2, NH2. So this is the urea, right? So urea relatively non-toxic compared to the ammonia, about 10,000 times less toxic. If 10,000 times basically means that if, let's say, if we want to carry one ammonia, then now I can carry 10,000 of the urea. So it means that I can transport them in the higher concentration. Okay. So urea diffuses from the liver cells into the blood plasma. As the blood passes through the kidney, the urea is filtered out and can be excreted. Okay. So with this, I have done for the first part. Okay, the excretion.